Okay, thank you, Mr. Chairman. I need both? Oh, sorry. Okay. All right, I must start off by thanking the organisers for this invitation. It's a great pleasure to be here, my first trip to Israel. I'm quite astounded that it's 3 years since we last convened in Canberra. Despite making me 3 years older, it has come around very, very quickly. But what I am excited about is what I've seen at this meeting, the enthusiasm, the diversity and the excitement around root research and where it's going. So I think that the field is in very good shape for the future. What I want to do today is give you an overview of the root soil microbiome. I've called it the other half of the hidden half in theme of the meeting. I'll give you some examples out of the literature. I'll give you some examples out of my own laboratory. But most importantly, I want to just sort of highlight some of the challenges and what the future directions are in this area. It's very complex. It's an exciting area. It's stimulated by the fact that we've heard about the human gut microbiome and how much interested in that. Well, the same applies to the plants. But there is a lot of hype around the area, and sorting through that hype and coming up with sound links between the microbiome and root growth and plant growth is a real challenge. I just acknowledge my co-authors, whom I will draw on throughout the talk. So, the benefits of below-ground biology, well, it's not new. We've known about soil microorganisms living with plant roots for decades, centuries. Plants don't live in isolation of the microbial communities that they associate within soil. They haven't evolved separately from the microorganisms, so the two have co-evolved together. So we should expect that there is intimate relationships between microbes and the plants that they associate with. And throughout the literature you can find all sorts of examples of plant growth promotion. We heard about symbionts and endophytes, rhizobia, mycorrhizae, a lot of beneficial associations, but also some detrimental associations as well. It's a constant battlefield out in the rhizosphere between pathogens and roots, and microorganisms that mediate that battlefield and provide suppression in soils. My own interests are in nutrient cycling around roots. I've spent now 30 years looking at phosphorus and nitrogen in the rhizosphere. And in the last part of my talk, I'll come back and try and connect some of the observations that we make about the microbiome in the first part of the talk and how we might use this in the future to better understand nutrient acquisition and availability by plants. So the root microbiome, or the soil biota, is very, very complex. It's not just bacteria, fungi, archaea. We tend to focus on them. They are the dominant players in terms of many of the processes, but it's a complex interaction of them with bigger organisms that form quite an elaborate network or ecological network below ground. Collectively, we can call it the soil metagenome, more simply the soil microbiome. And today is about how that soil microbiome interfaces with the plant genome, because they do interact with each other, they do talk to each other. And collectively, we can consider that as the holobiome. So it's the collective genes and the processes of the plant, combining with those of the microorganisms, and the functional contribution between the two that can give us what we could call the extended phenotype of the plant. It's complex, it's many compartments, and it's many organisms involved, and unravelling that complexity is clearly a major challenge. So in terms of capturing the benefit, this is fairly historical, this slide. We can talk about beneficial microbial communities. We know microorganisms in soil respond to practice change, to crop rotation. We know we can develop disease suppressive soils through crop rotation. The responses are evident. It's easy to document it and see it and say that it's due to a microbial or a biological response in soil. The challenge is how you actually identify and put a fingerprint, if you like, on what is a beneficial microbiome. Because if we could do that, perhaps we could then get greater advantage in the future from a management point of view. Or the flip side of the coin is using beneficial microbial inoculants. I mentioned rhizobia, mycorrhizae. There's been a lot of historic interest in diazotrophic nitrogen fixation, phytohormones to stimulate root growth, phosphate solubilisation. Clearly, in 100 years of literature, there's demonstrable success. We can all demonstrate success with microbial inoculants under controlled conditions. The real issue is the complexity in the real world of getting those associations to work in a way that can be reliable if we want to use it in agriculture for benefit. So it's demonstrable, but its widespread application is limited by inconsistent performance. And collectively in both of those, even though they're completely opposite in how we consider the microbiome, if we can understand the microbiome, how it assembles, how it localises with the root, how it interacts with the root, then our chances of success in both of those areas will be greatly improved in the future. Now I put this up because one of the things that has driven microbiome research, it doesn't matter whether it's gut microbiome, plant microbiome or wherever we are, is the new toolbox that has become available in recent years, the last 2 decades, particularly in the last decade with high throughput sequencing. There's 3 points to this triangle. I call it the omics triangle. Firstly, we can look who's there. We can do community structure. That's shown in green. We do that really well. It's really easy to do now. High throughput sequencing, we can look at 16S genes in bacteria. We can look at 18S intergenic regions in fungi. We're really good at describing what's there. No problems there. The real challenge is when you move to the other points of the triangle about the potential function or the in situ function. So just knowing who's there doesn't often help you to know what can they do or what are they actually doing. And when we're trying to link that with a trait or a response in plant response, and I'm interested in plant nutrition, as a plant nutritionist, we're pretty good at measuring process rates, at pools of nutrient in the soil, and understanding and making a prediction. If you tell me the phosphorus content of that soil is X, I can make a prediction about what plant growth I might expect from that information. I'm really good at describing the community over on the left side of that triangle as well. What I can't do is make a prediction between what I see on the left side of the triangle and what I want to achieve in my plant growth response. And later in the talk, I'm going to try and fill in some of those holes around metagenomics and targeted gene PCR. Of course, roots are complex. I don't need to tell this audience much about that. In soils, there's root types, there's age, location, compartments, different types of roots, nodal roots, seminal roots. Just listening to the talks over the last couple of days, we have enough trouble understanding what is the functional difference of a nodal root to a seminal root and what does it contribute to plant growth. Well, the same applies to a community of microorganisms that want to live around that root. We've done a lot of work in recent years looking at the role of different roots and what they do to microbial communities. I'm not going to talk about that today, unfortunately, but I can say that the microbial community that develops around a seminal root is very different to the microbial community that might develop around a nodal root. The community that develops around a root tip is very different to the community that develops further down on the base of the root. So spatially, it's very, very complex. In my lab at the moment, we're trying to work out what is the role of specific exudates in shaping that community. And one of the things we do focus on in a strength of the group that I represent is the role of organic anions and what role they serve for plants. We're also looking at the role of organic anions and how they mediate the microbial community. Much of that work is being done by Aki Kawasaki in our lab, and unfortunately I won't talk about that today. The other part of the roots is they're structurally diverse. We've got different compartments in the root as we go from the epidermis into the core of the root. As well as being structurally divergent, we also know that the environment around a root is very, very chemically diverse as well. All sorts of exudates, signal compounds, signalling and communication, microbe to microbe, plant to microbe, microbe to plant. It's a very, very complex world out there involving many different signals. I can spend 40 minutes probably on this slide alone and what it means, so I'm not going to spend a lot of time other than to summarise that we know root exudates have a big role in proliferating and providing an environment to stimulate microbial growth around the rhizosphere. As you move up, you have more specific signal compounds like flavonoids, strigolactones, that communicate with the mycorrhizae, the rhizobia, etc. So there's a lot of information going on there. We move up to the plant microbe communications and how a microbe then associates with the rhizoplane of the root. There's a whole cascade of information that happens inside the plant. Hormonal pathways, induce systemic response, I'm hoping the next speaker might pick up on more of that in his talk, so I'm not going to go there now. But it is a very, very complex pathway, and we know that microbes that are associating intimately with the root have to understand or have to communicate through those pathways to be a successful coloniser inside the root. To illustrate the complexity, I put this slide up. It's a slide that was generated by Michelle Watt when she was at CSIRO working with Margaret McCulley, because this picture tells a 1,000 words. I heard that from somebody else yesterday. I think it was Hans. And this just shows you the complexity of the environment that a microbe's got to deal with and understand. I was thinking about it this morning. It's a bit equivalent. If you're a 2-micron microbe sitting in a xylem vessel inside of that root, it's a bit like me sitting at a cafe in Jerusalem in the old city, sipping a glass of water. I've got the old city wall around me. Beyond that, I've got the suburbs, I've got the hills. It's equivalent to me sitting there in all of Jerusalem and expecting the city to be exactly the same in the environment that I'm sitting in. It's really quite different, and that illustrates the scale of what a bacteria would experience if it's sitting within that environment. What we can do, though, is we can look at different root compartments, and for convenience, 4 different compartments here: the bulk soil, the rhizosphere soil, the rhizoplane itself, and inside the root, inside the epidermis, inside the exodermis, inside the endodermis, known as the endosphere. And these are very distinct compartments. To illustrate the importance of these compartments for microbial communities, I take this example from a paper published by Edwards in 2015. A glasshouse experiment. They took 3 different soils, 6 genotypes of rice, looked at the 4 compartments and where the microbes are associated. And this is just a PCO multivariate analysis, and quite clearly, across 3 soils, quite clearly you can see the strength of the compartmentation there in differentiating these communities from the bulk soil, the rhizosphere, the rhizoplane and the endosphere. And you'll see the strength of that compartmentation is even stronger in this case than the 3 soils. There's actually 3 soils indicated there by symbols, and within each of those coloured symbols is also 6 genotypes. So the strength of compartmentation is stronger, much stronger, than the strength of the genotype effect. We can look at the effect of that on richness. So species richness is just a count, if you like, of the number of species being identified through the high throughput sequencing as OTUs, operational taxonomic units, but that's analogous to a species. And we can see a dramatic decline as we move from the bulk soil and enrichment in the rhizosphere, but then a very tight specificity of selection as we move into the rhizoplane and into the endosphere. That's probably better shown by these rocket or comet tail plots, where the coloured symbols represent a OTU or a species that is either specifically enriched or specifically depleted when it's below the line compared to what its existence was in the bulk soil. And you can see as you move from the rhizosphere to the endosphere, there is a progressive enrichment of organisms and a very, very strong depletion of organisms. So differentiation to exclude them. Very, very strong selection process as you move from the bulk soil into the endosphere. And you can start to put up some Venn diagrams and pie charts, just to, if you highlight the blue there, only 118 organisms in this particular example out of many, many thousands, out of 27,000 different species here, or OTUs, are specifically enriched inside the root, whereas over 1,200 are specifically excluded from being inside the root. And you can take that down to another level and we can convert those OTUs into species. And when you see the coloured dots here, basically we're seeing a very strong enrichment of the proteobacteria, the firmicutes and the bacteroides inside the root as you move through those compartments. We've done a very similar study in my lab, except we've done it in the field, and we looked at 2 genotypes of wheat in the field, and we sampled over 6 time points over 2 seasons. We were trying to get an understanding of how important this complexity is and how it operates under field conditions. So we had 2 cultivars and a fallow soil where we interchanged the cultivars over time through rotation, and we looked at 3 compartments. We looked at the bulk soil, the loosely bound rhizosphere, and those that are tightly bound. So we combined the endosphere with the rhizoplane. You can separate them out. Methodologically, it takes a bit of effort, so for convenience, we combine them. When you do that, and this is just another multivariate analysis, you can see that the dispersion or the diversity of the organisms are much, much higher on the rhizoplane than they are in the rhizosphere or the fallow soil, indicating a much larger dynamic of the community over time, and this is over 2 years. And you can see there's actually also some sort of consistency in what happens in year 1 and year 2. If we ascribe the variation through a model and look at the sources of variation, the localisation on the root or the compartment is by far the biggest describer of the variation that we see in that experiment. Crop development is the second most important. So there is an evolution or a growth of the microbes on the crop over time. We did it at 2 paddock locations. There was significance there, but it wasn't the major driver. Crop sequence was only a small variant in driving the variation, and most of that variation came of the wheat after fallow rather than wheat after wheat. But most surprising was only a very small amount of the variation could be explained by genotype, only 1%, which was quite surprising, but admittedly we only had 2 cultivars in there. That was supported with high throughput sequencing. The localisation was particularly important. And so then we can have a look at the community and look at it at the highest level, at phylum level anyway, and we can see clear differentiation when we assign phylum distribution. 80% or so fall into those 3 major phylum I mentioned before, particularly the proteobacteria. We can see clear differentiation between the rhizosphere and the rhizoplane communities, and we can see an evolution of the community of time as it evolves with root growth and crop development. If we just break that down further, and I just take a single time point here, the rhizoplane community at one time point, we can break that down to class, order, genus. I'm not going to go through that because it's so hard to interpret, but to say that by the time we get down to genus level in this particular experiment, we had 140 genuses of bacteria that were either up or down enriched significantly different. There's almost a programmed composition, as we found over the 2 years. It tended to repeat itself, which was really quite interesting, but surprisingly, as I said, we didn't have a genotype effect. So to explore that further, we went back and picked out 22 modern wheat varieties and grew them in a controlled environment, and we just looked at the tightly bound community in this case. This 22 varieties probably represents about 90% of the germplasm that we'd find, or the genetic variation we'd find across Australian wheat cultivars at any time. When we looked at the communities, both of the fungal 18S community and the bacterial 16S community across there, 22 cultivars, neither for bacteria or fungi did we get any significant difference. We found no effect of genotype on the community structure at the operational taxonomic unit here for either bacteria or fungi. If we look at that in terms of a class structure, you can sort of see how similar they are. You're more or less looking here at the core microbiome at the class level, both for bacteria major classes and fungal major classes. Again, no significant difference here across the class association across 22 modern wheat varieties that we're using in Australia. We did find some interesting bits of the data when we analysed it a bit harder. We found significant difference in Shannon's diversity. So there is some differences, but understanding the nature of these differences and certainly trying to understand the functional natures of these differences is very challenging. What we did do though, if we pick out a small cluster of 7 of those isolates, we have very good genetic information. They actually form 7 of the parents of a large mapping population that we have at CSIRO. And those 7 cultivars share between them 23,000 SNPs, and we could then look at the genetic similarity of those cultivars based on their percentage of shared SNPs. So any pairwise combinations of any 2 of these cultivars give us between 49% and 62% similarity between, or the genetic relatedness of these parents. When we plot that against the average pairwise similarity of the microbiome OTUs for bacteria or fungi against the wheat genome, we find no apparent relationship, confirming that really we're just looking at a single core microbiome across 22 cultivars with very, very weak, in this case, genotype effect. I just want to compare that to a literature example, though, by a French group published a few years ago, where they looked at exactly the same analysis that I did in the previous slide, but they looked at microbiome, in this case a bacterial microbiome, of 4 divergent maize lines, teosinte, which is a parent of modern maize, sorghum and wheat. And they found, like we would expect, quite different community structures in the rhizosphere for these different parents, And when they plotted the dissimilarity of the microbiome community versus the plant phylogenetic distance, there is a relationship. So we are confident that genotype can drive microbiome community structure, but within the modern wheats that I showed you, we didn't find that relationship. So the second part of the experiment, we then went back with those wheat cultivars that I mentioned before, but we took a wider germplasm or diversity panel of land races and relatives. So we took 16 land races of modern wheat, three modern bred wheats, and a range of progenitors and related species, wild relatives. And when we do that, we now do find quite different microbiome structure. And here I've just clustered by 3 colours according to the relatives, the land races and the varieties. And you can see that we do now start to see quite different community structures when we bring wider genetic diversity into the wheats. We can do the same thing that we did before. We can break it down into the different families, species and orders. I won't spend a lot of time on it, but 30 families accounted for 90% of the variation that we saw across these wheat lines, compared to those same families that counted 90% of the variation in the wheats and the presence in the wheats were only present in 40%. So there's a strong enrichment for these particular families. There is differences across the genotypes here, or the genotype groups, and we start to find some variation in them. So then we can start to break that down into genotypes or to OTUs that are specifically represented in either the land races, the relatives or the varieties. We can start to develop what is a core microbiome across the Tritiaceae. We can start to look at what are the specific OTUs or species that are being selected out from the soil. It's leading to the concept of a core microbiome, but the most important thing we need to do is to try and associate that with functionality. We can do the same with the fungal microbiome, and interestingly here you'll see that the Glomeromycetes, which are the mycorrhizal fungi, strong presence in the 3 lines or major groups there of wheat, hardly any presence in soil. So there's one very clear example of the selection power of the genome in pulling out specific groups of, in this case, mycorrhizal fungi. And we can do the same here for the fungi. We can start to look at what does constitute a core fungal microbiome, and the bigger question is, what are the key functional groups? So to summarise to this point in the talk, I just want to sort of emphasise that despite a lot of what I read in the literature at the moment about we will use cultivars, we will use genotypes to select out more beneficial microbiomes, I'm not that confident that we are in a powerful enough position to say that we can do that with confidence. There is a lot of hype around that at the moment. Sure, there's big progenitor effects. If we can understand the functional basis of the difference between the progenitors and what they might bring to our modern varieties, there is a potential to bring other parts of the microbiome. Plant species has a large effect. I didn't have a lot of detail in that. What I didn't talk about today, though, was management, soil and geography. They are all major drivers of microbiome structure in soil. So to try and put that into a bit of a model in terms of what's happening, out in the bulk soil we have high level of diversity, complexity. We have attraction, proliferation and enrichment in the rhizosphere, particularly driven by sugars and signals. We then go to very high selection and specificity as we move into the internal part of the root, into the endosphere. We move from a system of having generalists with that high level of functional redundancy that we've known about in microbial ecology for many years out in the bulk soil, to a system where we have very much specialists inside the root and how they interact with the plant as endophytes. We move from a system of high diversity richness to one of high selectivity based on signalling and host interaction. We can also identify some of the major drivers for how that microbiome is assembled around roots, where in the bulk soil, soil effects like soil pH, organic matter, climatic factors have a big bearing on the community composition, on the microbiome structure, whereas when we move to the plant effect, it's around the species and the ancestors like I showed, and I still keep a question mark on the cultivar because we've still got a lot of work to do if we think that we can use superior cultivars to simply select more beneficial microbiomes. So what are some of the key challenges we face right at the moment? One of the real challenges is we need to convert what I've shown you in the last 10 or 15 slides, where we can describe the system. We've got to be able to move from the structure of the community to its function. We need wider consideration of some of the interactions. The spatial and temporal interactions in the rhizosphere and the microbiome structure is enormous. We need to look at things in networks, and we're starting to do that now. Rather than looking at bacteria or specific groups or classes that are going up or down, we need to look at it in a network analysis to try and understand how that works. We need smarter learning. Perhaps machine learning is being talked about to actually better understand how that operates. Ultimately, though, we need better prediction. We need prediction to be able to move from the descriptive microbiome to the functional microbiome, and of course its interactive microbiome with plants. So it's not an easy task to move from the functional microbiome with crop performance. What's the best indicator we have for a functional microbiome at the moment? I would say it's the performance of the plant itself. It's still probably our best indicator of when we have a beneficial microbiome. So we need trait association with our microbiomes. We need to be able to associate the microbiome that we measure and see and be able to link that with a functional or desirable trait that we're trying to link it with. So can we develop a more favourable microbiome? Yes, I think we can. There's still work to be done on host genotype selection, and I'm going to show you one or two examples where I think there is positive progress for that for the future. By better understanding the microbiome, as I said in my introduction, we will be able to deliberately manage systems to generate more favourable microbiomes for our systems, and it will improve our smart deliverables. I believe it will be able to better give inoculants and endophytes to plants if we know the system that we're dealing with, the environment they've got to live in, the processes that they've got to overcome. So in the last 5 minutes or so, 6 minutes I think I have, I want to talk about how we might better link this community structure, the descriptive microbiome, to the functional microbiome. And I showed you this slide before, a challenge to move from structure to function. I'm going to give 2 or 3 examples of work that I'm involved in around metagenomics and one around targeted gene qPCR, where we're actually trying to bridge that gap between our descriptive microbiome and our plant response, or our phenotypic response for the microbiome. The first one is around phosphorus availability in the rhizosphere. This is a complex slide, but it describes the interactions between plant and microorganism around phosphorus availability. Phosphorus in the plant, phosphorus in soil, whether it's in solution, whether it's in the solid phase, organic, inorganic, and some of the processes that are involved in this complex interaction of phosphorus mobilisation, mineralisation, the conversion and making it available for plant uptake, and the response to plant exudates, phytostimulants producing roots. So it's a complex story, and I won't spend a lot of time on that, but what I will do is pull the story apart to rather than be process focused, to look at some of the genes now that we can start to unravel that are involved in major parts of that pathway. Genes like phytostimulants, I've seen on a lot of posters, the ACC deaminase. We now have really good handle on this gene and being able to measure it quantitatively and what it means and what organisms that it's associated with that can then associate with plants to stimulate plant root growth. Phosphate uptake, I'm sure we're going to hear from mycorrhizae people around the interplay between phosphate transport genes from the fungal partner to the plant. We now have some good metabolic genes for phosphate solubilisation, particularly around organic anion pathways. But one of my particular passions is the genes in microorganisms around phosphatases. I've worked on these in the rhizosphere for many years: alkaline phosphatases, acid phosphatases, and phytases. A very, very broad collection of genes we know are critical and important for mobilisation of phosphorus from the organic phase into the inorganic phase to make it available for plant uptake. And the example that I'm going to illustrate is some work by Andy Neal, who we're now working with at Rothamsted, where Andy did some metagenome sequences across 3 of the long-term plot trials at Rothamsted. He generated between 230 million to 580 million reads per sample. He then binned, and they were just shotgun reads that were then binned according to the functional gene that he could identify them against. And when you do that and you normalise that information back against housekeeping genes that you also target in the metagenome analysis, you can indicate that if you take FOD in the middle of the top, that we expect around 50% of the microbiome community in that soil has a FOD in it. Compare that to a phytase gene, 3 classes of phytase genes, we find that between 1 and 2% of community members actually have a phytase gene in. So we can actually start to get an understanding of what are the predominant genes that are in the environment. And if you look at a gene like Phoa, he found significant differences between a long-term fallow and an arable or a grass environment. If you go back to FOD and you just do a phylogenetic tree of every FOD that you can find on the NCBI database, it's a pretty complex story. It's lost in a lot of noise about information around specific sequence identities that you get a tree like this, just plotted off the database. But if you then plot, for the Rothamsted plots, the predominant FOD genes, you find they actually very, very clustered. And you'll see there's 2 major bubbles. The cluster 1, which is the isoform or the form of the FOD that Andy is typically finding in the Rothamsted soil. So there's actually a very, very strong selection for that particular isotype of FOD in that soil from the metagenomic information. If you go to cluster 2, that is the predominant isoform of that gene being found in the fallow. There's been no plants for 60 years in those plots. So there's a very strong plant microbe association here being driven by long-term effects. What's really exciting about this is we're working with Andy now. We're taking this information to as many metagenome data sets as we can across the world. What we want to try and elucidate is, are there absolute key functional FOD genes that represent in other environments? Can we get away from all the noise that we've generated in molecular biology over the years to find out what really are the key genes in the environments? And we're doing this not just for FOD, we're doing this for a whole range. We've got about 6 or 7 genes that we're dealing with carbon, nitrogen and sulfur cycling to try and find a real core of genes that are associated with the environment. The second 2 examples I'm going to give are around nitrogen cycling. Same story here, it's a complex slide, I apologise for that, but this basically summarises everything we know about microbial interactions in the rhizosphere in relation to nitrogen cycling. Whether it's the plant nitrogen and the forms and the conversion of nitrogen in the plant, or the processes in the rhizosphere, the soil solution, or with the soil organic matter, particularly around the nitrification of nitrogen, that is the conversion of ammonium to nitrate, the denitrification processes, that's the gaseous loss of nitrogen from the system, or the interaction with root exudates, including biological nitrification inhibitors, which we know come out of the roots of many plant species. Again, not worried about the forms and the fluxes and the pools of nitrogen. We can actually break this down from a microbial point of view to start looking at what are the key genes that are involved in different processes. For example, NIFH, a gene that we've known about for a long time, a very important gene for nitrogen fixation. Apart from the rhizobia NIFH, which we know is in nodules, we also can characterise NIFH present in diazotrophs. And these diazotrophs may be associated with the endosphere, living inside the plant, or diazotrophs living out in the rhizosphere. For nitrification, we have a very good handle on the ammonification A gene, which is the key gene involved for conversion of ammonium to nitrate, and we have good handle on various genes in the pathways for denitrification. To put these into some context, there's some work. This is work by one of my co-authors, Gupta Vadakatu, and Gupta's been particularly interested in diazotrophs for a number of years now, and he published quite a significant paper, I thought, a few years ago, looking at NIFH. Okay, I'll be about 2 or 3 minutes. 2 minutes, I think. So I've just got 5 minutes. He looked at the genetic variation of NIFH across these grasses that some are active, live in low nitrogen soils. Big differences in community structure around NIFH for these 4 grasses. And once again, very strong compartmentations. Shown in the middle there, roots and soils quite different. He also found that specific isoforms of NIFH were heavily enriched in the endosphere as opposed to the rhizosphere. And understanding that enrichment and the functional contribution of those variants of NIFH to the nitrogen fixation that he could measure in those grasses is proving to be quite important. We've recently extended that to look at NIFH abundance in the rhizosphere of cereals, and just showing here very quickly, to our surprise, and we don't understand the mechanistic behind this, but big difference in the abundance of NIF-H enrichment in a barley rhizosphere compared to a wheat rhizosphere. We don't know the basis of that at the moment. It represents a great potential for biological nitrification in our cereals. And one of the things we're doing right at the moment with the landrace information that I showed you on the wheats is going back to look what sort of variation we have for NIF-H genes in our wheat landraces. Very, very quickly, 2 slides on this one. A paper from a postdoc in my lab where he has just recently plotted this over 3 land uses over an ecotone system, measuring nitrification rate and being able to relate that to the abundance of the MOA gene. To get this sort of relationship in microbial ecology in the real world is a challenge, and it's always good when you see a positive relationship, a bit of noise around it, but we get a positive relationship. What's important about that is that we know that the abundance of the bacterial MOA gene for nitrification is heavily suppressed by species that can produce biological nitrification inhibitors like Brachiaria as shown. If we go back to our land races of wheat, this is work from Catherine O'Sullivan in our lab, where she screened those land races for variation in BNI, and just shown on the left of the graph there is 5 land races of wheat that have very, very significant BNI release from their roots. Compare that to Brachiaria in the middle of the plot, compare it to 2 wheat cultivars further over in the plot. What we can show is that when we look at the BNI capacity of those plants, they have tremendous capacity to change nitrogen cycling in the rhizosphere, where we are seeing an effect here now of BNI from land races of wheat having a significant impact on whole soil communities and manipulation of the microbiome. So even though I said that cultivars are difficult to choose, there is potential that we can use specific genotypes to manipulate the system. So just to close, just a few concluding comments. We do have extensive understanding of the microbial recruitment process and how it assembles. The challenge remains is how we move to a functional microbiome, how we move from this concept of a core microbiome, but in terms of being able to describe it in functional groups or functional genes. We need to consider the microbiome and its interaction with the bigger system that it works with, and preferably or ideally we need to link that microbiome with plant traits. Is there opportunity to capture benefit from a more favourable microbiome? Yes, I think there is. The last example I showed you is an example where we could start to use genotypes to select more favourable microbiomes, such as in the BNI example. As I said before, we can use agronomic management interventions. The more we understand about the microbiome, how it associates, how it assembles, how it functions, will give us a better chance to capture those benefits. And lastly, as I said before, we can develop smart deliverables for our inoculants, particularly our endophytes, as we learn more about endophytes, to capture benefit of them, but we need to understand how microbiome structure and function to be able to achieve that. So thank you very much.